All right. Hello, everybody. So now we're going to start to talk about gene regulation in eukaryotes, aka, and you thought the lac operon was complicated. So what are we going to talk about in this first lecture? There's going to be three sort of mini lectures that we're going to do for this portion of the class. And this first one just covers the very first part of chapter 15, regulatory transcription factors. So let's look at some gene regulation in eukaryotes and see where we might be going. So last section, we talked about gene regulation in the lac operon. That is, of course, a prokaryotic system in bacteria. Eukaryotic systems, such as us, especially multicellular eukaryotic systems, derive a lot of benefits from regulating the expression of their genes for lots of different reasons. A couple of examples include things like what the lac operon is for, responding to changes in nutrient ability and allowing you to use different things for nourishment. You can also respond to environmental stresses by changing the expression of your genes. But we are much more complicated than prokaryotic organisms, and that is reflected in the complexity of our gene regulation. So why is gene regulation so important? Well, if you start to think about how complicated you are as an organism and the kind of things that your genes can do, right? One of the big things that a lot of people don't really think about that your genes do is they basically drive how you are patterned as an organism during development. What do I mean by that? Well, as you know, when you first came into existence, right, you were a single cell big, and that was it. And then that cell divided into two, which divided into 4 cells, which divided into 8 cells. And gradually you got bigger and bigger and bigger, right, in utero. While that process is going on and you're growing as an organism, decisions have to be made. Initially, those decisions are things that are as simple as which part's going to be the head, which part's going to be the feet, which part's going to be the dorsal part, which part's going to be the ventral part. In other words, the back and the front. And so those decisions have to be made. And then going on from that, you have to make decisions as to, you know, what cell is going to be part of your finger, what's going to be part of your bone, that kind of things. And so those decisions are governed by gene regulation, controlling which genes are expressed where. So development is driven by changes in gene expression. And then, of course, you've got all the cells in your body that do all these different jobs. And as you know, that is driven by changes in gene expression too. And that's because all of your cells are identical genetically. So what makes the difference between a liver cell and a kidney cell is simply which genes are expressed in those different cells. Here's a couple of examples. This is an example of a change in of how genes can regulate development. Here are 2 mouse embryos. What you see along the sort of spine of these embryos is black stripes. That is a gene that is really important for somite formation, which is the muscles along the back. We have those muscles. Somites are the muscles that you eat if you eat a fish. Stripes along the back of an organism. You're looking at a normal animal in panel A here, and panel B is a mutant animal that has its gene expression screwed up. And so what you can see is that those nicely defined somites in part A now look all messed up. And that's because there's a gene TBX6, which is mutant in that organism. That is a transcription factor that controls transcription of an important gene, therefore, which is shown in black. When that regulation is messed up, you get messed up somites. And in fact, this organism is phenotypically very screwed up. In an adult or fully developed organism, as you guys know, the subset of genes that are expressed in a cell that, determine what it does. Of course, all that is determined by tissue-specific gene expression. So, gene regulation, very important in eukaryotes. Because of that, it's phenomenally complicated. So remember what we're talking about when we're talking about gene expression and what that term refers to. Gene expression is the journey from DNA information to a fully functional protein, as shown on the left half of this slide. So DNA, mRNA, functional protein. That process is called gene expression. On the right, you see a picture of a cell with that same process, right? Begins with a gene, DNA, ends in a functional protein. And what you see as you go down these arrows is every different stage in that process. Gene is transcribed into pre-mRNA, which is spliced and changed into messenger RNA, which is translated into a protein, which eventually becomes a functional protein. And what you can see here is a short description on the right about how each one of these things is regulated. So you can see that there are many levels of gene expression regulation in eukaryotes. Essentially, if you can think of a place where you could mess around with gene expression, it's going to be regulated in eukaryotes. So it's very complicated. We, however, are going to focus our study on mostly what happens at the transcriptional level. So what's happening right at the start here. And that's because that is where the most sort of fundamental decisions are made whether or not to express a gene. Because when you've made the decision to transcribe a gene, you've made that decision, right? So there's a sort of on and off regulatory step at the start. Now, to look at what happens in that part of transcription, of gene expression regulation. Sorry. We need to start with a look at things that are called transcription factors. What is a transcription factor? A transcription factor is, it won't shock you to hear, a factor that influences transcription. The word factor is what people kind of used to call proteins a long time ago. And all transcription factors are proteins that affect transcription. And normally, every transcription factor, really, its end goal, they do it in lots of different mechanistic ways, but their end goal is to influence the ability of RNA polymerase to transcribe any given gene. And there are sort of two main types of transcription factors. The so-called general transcription factors, which are required essentially at every single gene. So they're required for the binding of RNA polymerase to the core promoter of a gene, and they help it get going. They're just necessary for transcription, period. So any gene requires those. So we're not that interested in those when we're talking about gene transcription regulation. What we're more interested in is regulatory transcription factors, which are much more interesting because they are very specific for certain target genes. Just to let you know how important transcriptional regulation is, if you look at the entire number of genes in the human genome, roughly 22,000 or so, 2 or 3% of them encode transcription factor proteins, which is a lot of genes. And that's because they're really important to how you function as an organism. So what do these regulatory transcription factors do? Well, essentially, they work in very similar ways. They recognize cis-regulatory elements located somewhere near the core promoter, somewhere not. A cis-regulatory element, if you remember from your lac operon days, is something that's on the same piece of DNA as the gene that it regulates. What most regulatory transcription factors do is they bind in a sequence-specific manner to DNA and regulate a gene in that way. So they literally bind to DNA at a very specific sequence. That's how they can be specific for different genes, because DNA sequence tells them where to sit on the DNA. And then they control the expression or the transcription of their associated gene. There are different types of binding sites that these kind of things bind to. And some transcription factors will upregulate or increase the transcription of a gene. Generally, those are called activators. They bind to a set of DNA sequences that are called enhancers. Now, enhancers are all different sequences, but that's the term for a piece of DNA that binds to an activator and allows it to upregulate gene transcription. The flip side are transcription factors that downregulate or inhibit gene expression. Those are normally called repressors, like the lac repressor is the example that we already know about. And the sequence that they bind to in eukaryotes is called a silencer. So that will be similar to the operator sequence that specifically bound the repressor in the lac operon. So on one hand, you have activators that bind to enhancers, turning genes on or upregulating them. On the other hand, you have repressors that bind to silencers and downregulate genes. And those are sort of the two general types of regulatory transcription factors that we're talking about. Please note that you kind of know this stuff from the lac operon, right? Very similar. You had a specific DNA sequence that bound to a protein that then, influenced the transcription of the operon. It's all the same in eukaryotic gene expression. The same concepts are at play. It's just more complicated. So here's a classic sort of model for gene activation. As you can see, there's an enhancer on this piece of DNA. It is binding to an activator protein. This would be the transcription factor, the regulatory transcription factor. And then just downstream from that, we have the core promoter, which binds to RNA polymerase, which is kind of this dark brown colored protein. And general transcription factors are also present here. They're required again at every single gene. But it is the binding of this activator protein to this enhancer that causes this big complex of proteins to form, turning on transcription. So this is the transcription factor that we're talking about, binding to the enhancer. The flip side, as I said, was gene repression, which is based on repressor type transcription factors, binding to silencer type DNA elements, as you see on the bottom here. And then again, of course, we see a core promoter, but notice the difference. This silencer binds to this repressor, somehow conceptually speaking, shutting off this gene, inhibiting transcription of it. As I said, eukaryotic control, you know the general concepts behind this from the lac operon, but the biggest difference really there's two massive differences, but one of the biggest ones is combinatorial control. So in the lac operon, you did have some semblance of combinatorial control because you had the lac repressor, right? And remember, you also had the CAMP binding protein that bound to the DNA sequence next door. So you had two different things controlling the lac operon. That's combinatorial control. But in multicellular complex eukaryotic organisms, it's much more complicated. So activators and repressors will both have influence over a gene. There will be multiple activators, multiple repressors, that bind to different sequences. And those proteins themselves, those transcription factors, can bind to small effector molecules, kind of like allolactose. They can bind to other proteins to form protein dimers to affect gene transcription. They can be covalently modified by phosphorylation, things like that. So this is called combinatorial control, and it's way more complicated than the kind of control that you see in prokaryotes. But again, conceptually speaking, we're just talking about proteins binding to DNA sequences and influencing transcription. So transcription factors, there's a good few hundred of them in the human genome. They have a lot of similarities. The proteins themselves contain regions called domains. So you can have a transcription factor where part of the protein is involved in sequence-specific DNA binding. The other part of that protein could bind to a regulatory molecule, for example. Those two different parts of one protein are called domains. There's another word that are called, that is a motif, which is a specific domain or part of a domain that's very similar in lots of different transcription factors. So you can have, for example, a DNA binding domain or a DNA binding motif that is shared or very similar between lots of different transcription factors. And I want to give you some examples of those motifs and domains, because there's sort of four main types of transcription factors. So the first one I want to talk about is a helix-turn-helix motif. And what you can see here is that in blue is DNA. And remember, I said these transcription factors bind specifically to a DNA sequence. That's what a helix-turn-helix motif does. It's called that because it has an alpha helix, followed by a turn, followed by another alpha helix. And these parts of the protein molecule are involved in recognition with specific DNA sequences. So that's sort of the targeting part of a protein. And different transcription factors might have slightly different helix-turn-helix motifs as part of their structure, which allows them to bind to different DNA sequences. Another motif is a helix-loop helix motif, very, very similar to a helix-turn-helix. You guessed it, there's a loop instead of a turn. So here we have a motif that's made up again of an alpha helix that's a recognition motif. There's another one kind of crisscrossing it. And this time there's a loop involved in their structure. So that's what gives them this name, a helix-loop-helix motif. And again, in a transcription factor, you would have, a helix-loop-helix motif joined to another motif that regulates it somehow. A zinc finger motif is the third of the four we're going to look at. Zinc fingers are very common to, again, recognize different specific DNA sequences. They're called zinc fingers because they're a little bit more complicated than a helix-turn-helix, as you can see. They have a slightly more complicated three-dimensional structure. They got their name from the idea that they're sort of fingers that stick in between the helix of the DNA, as you can see here. And they require zinc as a cofactor to stabilize their three-dimensional structure. They're called zinc finger motifs. And last but not least, I mentioned that sometimes transcription factors can be dimers. They can be two proteins that join together to give you a functional transcription factor. Sometimes when they join together, what joins them together is something called a leucine zipper motif. Leucine is an amino acid, one of the 20 that make up proteins. And leucine zipper motifs are so called because, as you can see in this little cutout box, there's a amino acid sequence here, and sticking out from the side of it is leucine amino acids, and they literally close together like a zipper. And so the two different colored amino acid sequences here kind of zip together. And you can bring two proteins together to give you a dimer, a protein dimer. And that protein dimer then gives you this recognition motif that again binds to a specific DNA sequence. So these are the types of transcription factors that exist. And you notice that of all the 4 types, they all contain these motifs that recognize specific DNA sequences, which is going to decide what gene they're going to regulate. Because whatever this DNA sequence is that this zinc finger is recognizing, it will be important for regulation of a certain gene. Okay, so thinking about eukaryotic gene regulation, we think about transcription factors, specifically regulatory transcription factors. They're going to bind to an enhancer to upregulate a gene, or a silencer to downregulate a gene. We just talked a little bit about the specific 3-dimensional structures of those motifs that transcription factors contain. I just want to say a quick word about the DNA sequences they bind to, so enhancers and silencers specifically. These response elements, that's kind of a catch-all name for enhancers and silencers, because there are elements in DNA that bind to transcription factors to elicit a response. They have some really interesting characteristics. They are orientation independent. So it doesn't matter which way round that specific DNA sequence is. They normally act the same in either orientation. So they literally just act as places on the DNA that bind to proteins, and it doesn't matter which way round they are. Most enhancers and silencers, by the way, are relatively short. We're talking about 10 base, like a dozen base pairs or less. So pretty small bits of DNA. So that's kind of interesting. That was a surprise when we figured that out as biologists. And then also interestingly, I said that the combinatorial control means that a lot of transcription factors can kind of weigh in on the repression or either the downregulation or upregulation of a gene. The different transcription factors bind to different response elements because they're sequence-specific binding proteins. And the response elements, apart from being orientation independent, can also be positioned at really weird places compared to any gene they're going to control. So shown at the bottom of the slide here is a piece of DNA with some enhancers on it. And the actual core promoter is shown in blue here. And here is the introns and the exons of the coding region of this gene. And what you can see is that enhancers can be proximal to the promoter right next door, but enhancers can be upstream of a gene on the left. They can be downstream of a gene, sometimes by 100,000 bases, so a long way away from the gene that they're regulating. And they can even be within the gene itself, so you can find them within introns as shown right here. And these are all shown as enhancers, but equally these could be silencers. So these guys are, when you talk about response elements that bind transcription factors, they're generally said to be distance and orientation independent. So they can be either way, because DNA has an orientation, right, 5' to 3'. These guys can be located in either orientation and lots of different distances away from the promoter that they actually regulate. So that's it for the first part of this little lecture set. In the next part, we're going to talk about some of the mechanisms by which enhancers and silencers regulate the genes that they do. So I will see you for that next.